Hello, everybody. My name is Radwa Moanis. I am a PhD student in the research group of microbiology in the VUB under the supervision of Professor Evelyn Peters. And today I will give you a short a short overview about my research on microbial production of polyhydroxyalkanoates by thermophiles. As you all know, petroleum-based plastics are really important nowadays in our life, and they are used worldwide. But they fit in a linear economic concept, as they are produced from non-renewable fossil fuels that also cause increasing global warming, and the product, the plastics, is very difficult to be degraded in the environment, causing a lot of problems to the marine environment and also to our health. So finding an environmentally friendly alternative is a real must, and this alternative has to be bio-based and biodegradable bioplastics, and this is my work. The goal of my PhD project is to produce bioplastics from bacteria. The bacterial bioplastics is called polyhydroxyalkanoates, which can be abbreviated as PHA. PHA are polyesters produced by microbes and which are completely biodegradable. They are produced by numerous bacteria and archaea as intracellular PHA granules inside the cells, as you can see here. And they are produced in response to unbalanced nutritional conditions, as excess in carbon source and limitation in other nutrients as phosphorus and nitrogen. And also might be another environmental stress conditions, as for example, increase in salinity. These granules are reserved for carbon and energy, and then the bacteria can use it later. PHA can be classified according to the number of carbon atoms in the monomers. So they can be classified according into short chain length, where carbon atoms are less than 6 atoms, as the most common and popular PHA type, the hydroxybutyrate. And medium chain length, where the carbon atoms are between 6 to 14 carbon atoms, and long chain length, where the carbon atoms are more than 14 carbon atoms. The monomers can be linked together into homopolymers or copolymers. The copolymers composed of different monomers, as for example, PHB-CoV, so it's butyrate and valerate. And the copolymers usually have better characteristics than the homopolymers. What makes PHA interesting as bioplastics is its properties. So they are comparable to polypropylene plastics, and they are non-toxic. They can be produced from cheap substrates. They are insoluble in water, and most importantly, I think, they are complete biodegradable. As you can see here in the picture, the degradation of a plastic bottle just in 10 weeks. And also what's interesting about PHA is it fits in the circular economy. So we can start from renewable substrates, as for example, agricultural wastes and whey, and then produce the plastics, which can be then degraded, just produce water, carbon dioxide, and biomass. Then the plants can use these to reproduce the renewable substrates. Although these interesting properties, there is still some bottlenecks that impede the commercialization of PHA in a large scale. And the problem is the cost of the production. So the production cost of PHA is nearly 4 to 5 times as conventional plastics. And that's because of several reasons, and I will say some of them now. The high cost of fermentation and purification process, finding cheap feedstock, and also optimization of the properties of the PHA polymers. And when we thought about these problems, we found that the thermophiles can solve these problems. Thermophiles are heat-loving bacteria. They are the bacteria that grow optimally at high temperatures, over 45 degrees Celsius. And they can, for example, solve this problem by decrease the cost of fermentation by 2 ways, by saving energy costs and avoiding contamination. For the first one, as you know, for large-scale fermentation, there is heat produced from bacteria due to the metabolic reactions. And this heat can harm the bacteria or the production itself. So there's always cooling steps required, but it increases the cost. But when we use bacteria that optimally grow at high temperatures, we don't need this cooling step, so that the cost will be decreased. And also, as it grows at high temperature, there is not a lot of bacteria that can grow at these high temperatures, so we don't need the strict sterilization precautions or conditions that we normally do. So also this will decrease the cost. And also for thermophiles, a lot of them can grow on cheap substrates as agricultural wastes, and also some of them can produce copolymers with the better properties. So my research objectives is to study the PHA production in thermophiles, and here by thermophiles I mean both moderate thermophiles and extreme thermophiles. So moderate thermophiles are the thermophiles with optimal growth temperature between 45 to 65 degrees Celsius, while the extreme thermophiles with optimal growth temperature above 65 degrees Celsius. And also I'm aiming to optimize the PHA production and to have better understanding of PHA regulation in thermophiles. And finally, I would like to engineer PHA senses in non-PHA producers. So normally my workflow is to first do the growth experiments of my bacteria on different conditions, and then to detect the PHA produced. The routine way that I'm using in the lab is fluorescence-based techniques, and then doing chemical analysis of the extracted PHA, and finally do some genetic analysis to know the enzymes and genes involved in the production in these bacteria. So I will go quickly on the fluorescence-based technique because, as I mentioned, it's the routine way that I'm using in the lab. So I'm using Nile red stain, which is a fluorescence stain, which bind to the PHA granules to produce fluorescence. And this fluorescence can then be measured by the plate reader. So let's start with the first bacteria I want to show today. It's Thermophilus. Thermophilus is an extreme thermophilic bacteria. Grow optimally between 70 to 75 degrees Celsius. And as you can see here, this graph is from the plate reader. So normally in my experiments, I'm using a positive control. This positive control is Paracoccus denitrificans, which is a well-known PHA producer that produce high amount of PHA. And also I'm using E. coli as a negative control because naturally it doesn't produce PHA. And here we can see from Thermophilus there is some fluorescence, but much more less than the Paracoccus denitrificans. So we went to the next step to trying to do PHA extraction. I tried several methods and did the chemical analysis using GCMS, FTIR, NMR, and DSC, but all of them didn't give any results for Thermophilus. So from here we can say that no PHA accumulation was detected in thermos using chemical analysis methods. And also I combined these results by the bioinformatic analysis. And the same thing, we didn't find or we couldn't predict the classical PHA genes in Thermophilus genome. So we shifted our work to a moderate thermophilic bacteria, which is Shellagellella thermodepolymerans. Shellagellella can grow optimally between 50 to 55 degrees Celsius. And as we can see here, when we used several carbon sources, it gave high amount of fluorescence here. And also by comparing it to Paracoccus denitrificans, my positive control. And not only this, but also it can grow on xylose, which can be present in the agricultural wastes, and also glycerol, which is a waste from biodiesel production. So it's an interesting one. And not only this, but also by introducing valerate to the media as precursor, it can produce the copolymer polyhydroxybutyrate covalerate. So then also I tried environmental stress conditions as salinity. So I increased the concentration of salt in the medium of NaCl, and by increasing the concentration, the fluorescence, or I can say the PHA production, was increased. Although the growth decreased, but the PHA increased. Then by doing the a PHA extraction. So I did drying to my cells, and then trying to dissolve it in chloroform to dissolve the PHA. Then just by adding cold methanol to it, we directly can obtain the PHA, as you can see here. And then, as you can see in this image, there is PHA from different carbon sources, and finally also I could obtain the PHA sheet from the bacteria. And luckily this time, by screening the genome of Shillelagh thermodepolymerase, I could predict all the genes responsible or involved in the PHA production in Shillelagh thermodepolymerase. So now I can say some of the conclusions of my research. It appears that there is an upper temperature limit for the natural occurrence of PHA synthesis. So in extreme thermophiles, we couldn't obtain PHA, and it's not just for thermos, because we extended our bioinformatic analysis to nearly all the other extreme thermophiles, and we couldn't find the classical PHA genes inside. But in modern thermophiles, we can obtain PHA from it. So my aim now is to do engineering of PHA synthesis in extreme thermophilic hosts by enhancing thermostability of PHA enzymes using protein engineering approach, so that we can introduce it into the thermophilic hosts. And secondly, also we can say that Shillelagh is a promising PHA producer. So my next steps is to do to generate further biological insights to focus more on understanding the regulation of PHA biosynthesis pathway inside the Shillelagh, and also to use this information to optimize the PHA yield and the copolymer composition. And I would like to say that my research fits in a larger consortium of PHA research at VUB, which is combination between microbiology, organic chemistry, and material science. And also I would like to mention that Professor Nico van den Brand is also a member in CAPTCHA in FISC. So while at VUB we are working on engineering and initial characterization of new strains and biopolymers, we are seeking collaboration partners who can bring this innovation to a next level towards the application, as for example, process engineering, value chain development, and life cycle assessment. And thank you very much for your attention.